Translation is the very obvious part of molecular biology, and we're going to get through it fairly fast. And before we talk about translation, let's talk about what transcription is. Transcription, we've talked about it before. Transcription is when we just have a script being copied off from the DNA, and we call it the mRNA, and it's a coding RNA, the only coding RNA in our bodies as well, in our cells. And before we talk about translation specifically, we want to talk about tRNAs and how amino acids will bind onto the tRNAs, and we'll also talk about the structures of the tRNA. This is the first part of translation, so this is a very overall understanding of what translation is. So translation really is a process when we have amino acids coming together and forming peptide bonds between them to form polypeptides, and that's why it's called a polypeptide, because we have a lot of peptide bonds between amino acids, and this will synthesize an enzyme or a protein, and then this is going to just keep on growing, and really it depends on what amino acids are being made, depends on the mRNA and the specific codons on it. So you might be asking, okay, so we have the ribosome and we have the mRNA, so how the hell are amino acids, finding their way onto the ribosome, and how is this polypeptide being synthesized? So what's the entire mechanism behind it? So before we talk about the mechanism, let's get into the overall steps of translation. The first step is initiation. Initiation really just happens when we have the mRNA, and the mRNA has a start codon in it. The start codon will not code for any amino acids, but it just signifies the entire enzymes that are required for translation to bind to it. An example of it would be the ribosome, and we will talk about it in a future video. And the propagation is when we have elongation, and that's just really when we have the amino acid changes growing and forming more polypeptides or more amino acids, and the polypeptide is growing. And termination is when we reach the N-codons, which is on the 3' end, and that's when the entire mRNA is telling the ribosome that, hey, look, you've read me and you've completed the polypeptide, so it's time to dissociate. And this is how the ribosomal complex will fall off and all the enzymes will fall off. And then the mRNA can be further synthesized again, or it can be degraded depending on the cell's needs. But so here's the question that we want to ask, and we did talk about it. How are these amino acids finding their way onto these ribosomes? And that's happening through tRNA. tRNA is we talked about it before, tRNA really just stands for transfer RNAs, and this is an example of a non-coding RNA, which carries an amino acid to the, this is the tRNA's primary job is to carry the amino acid to the growing polypeptide on a ribosomal complex. So what is the structure of a tRNA? The tRNA also has a 3' to a 5' end. It's a RNA. It's a non-coding RNA. Excuse me. Excuse me. So we have if you talk about the structure of the tRNA, it's a stem and loop structure, and that's because if you notice, the whole structure of this complex here is stem and loop. And another thing to mention real fast is that the tRNA is synthesized through the RNA polymerase 3. And if you don't know what the RNA polymerase 1, 2, and 3 does, just go look at the previous video, and you'll understand what all the different RNA polymers do, and don't confuse them with DNA polymers, please, and that's discussed in that video. And the second thing we want to focus on when it comes to tRNA is the anticodon. The anticodon really is our main focus in this whenever we talk about tRNAs in biology, and that's because the anticodons are very specific to the codons on the mRNA. It's really, so if we have a mRNA strand, and the mRNA strand has a codon, we talked about the codons. We said 3 nucleotides makes 1 codon, and there are multiple experiments done on this that prove that 3 nucleotides, they're 1 codon. They code for 1 amino acid, and the whole process of how this code is recognized is through the anticodon. And the anticodon is just for the mRNA. And here's a question to ask real fast, considering the fact that we talked about structures before. We talked about we talked about structures, and we talked about quaternary structures, or sorry, tertiary structures, which is the internal reaction between all the elements of a polypeptide, and or excuse me, or it's a structure based on the tertiary structure is based on whatever is happening inside these this isn't the reason we won't call this a polypeptide is because it's not an amino acid, but the entire point that I'm trying to get at is that these chemicals or whatever or the nucleotides is composed of are interacting with one another, and this is forming a structure. But the anticodon, do you think that this anticodon has any impact on the entire structure of the tRNA? And the answer to that question is no, it does not contribute to the tertiary structure of the tRNA. And that's because these anticodons are reserved for the codons and just for the codons, and they will interact with just the codons on the mRNA. So that's a very important point that you should understand by now. The next thing we want to talk about is the amino acid. The amino acid, the specific amino acid for the tRNA, will bind to the 3' end. And we will get onto something called we're going to talk about the CCA, I believe, the CCA, the CCA bases over here, which will allow the amino acids to bind to it. And we'll talk about that in a second. There you go. I thought we didn't have it on this video, honestly, but that's fine. Okay, so here's a question to ask. There are 20 different amino acids. So we know that our bodies have at least 20 different amino acids. And what do you think? We have different tRNA components of the tRNA. So, sorry, what I'm trying to ask is there are 20 different amino acids in our bodies, and because of that, there has to be a difference between the tRNAs as well who carry them. So what are the biggest differences that inside a tRNA molecule compared to another tRNA molecule? So let's say that we had a tRNA molecule right over here that codes for valine, and let's say we had, one second, let me just duplicate this, like I could duplicate everything else on this planet. 1 second. So let's say we had two tRNAs, and don't be fooled by the size, it's just an example. If you had two tRNAs and this tRNA codes for valine and this codes for something else. So what is the difference? What is the primary difference between these two tRNAs? Because they both are very similar compared to the chemical, or the tRNA structures are very similar. So what makes these two different? So the anticodon will be the major difference between these two, and the anticodon really determines where this tRNA it will be able to attach itself to in the mRNA. So 60% of the body is similar from one tRNA to another, and the amino acid binding site to CCA 3' end is also also similar. So we're going to talk about how amino acids will recognize their specific tRNAs in a different video, but the highlight of this just really is that the anticodon is the most differential thing, and it's the most important thing when we talk about tRNAs in comparison to where they're going to bind at. So here is a tRNA and a specific amino acid. So amino acids can have let's talk about this for a second. So we have a tRNA right over here. This tRNA is designed for just one amino acid. There's only one amino acid that has the ability to bind onto it. But anything else, any other kind of amino acid, let's say this is a valine. The valine for this amino acid, the valine amino acid, will only bind to this tRNA. We won't have any other amino acid that has the ability to bind to this tRNA. But this means that tRNAs are specific to amino acids. However, amino acids are not specific to a tRNA, because if we remember, if we saw the coding, we have a chart which shows us what nucleotides code for what amino acids, and if you look in that, you will realize that there are different codons that code for the same amino acid, and because of that, we can have several tRNAs for a specific amino acid. Now here's a method of writing tRNAs. And if we have a tRNA by itself, and this tRNA is a tRNA that will carry the valine amino acid and doesn't have the valine on it yet. So this is how we write it. This is how it's going to be denotated on a test, or if you want to write it yourself, it's a tRNA VAL at the bottom over here. And if the valine is attached, the amino acid is attached to the tRNA, we just have to write val in the start. So VAL dash tRNA. Wow, that's just indicating the val in the start indicates that the amino acid is attached to it. So before we end this video, we want to quickly talk about the structure, the visual component of the tRNA. The tRNA obviously doesn't look like a neat structure like we see it over here. It's a 2D structure, and if it were a 3D structure, it'd be something like randomly. It would be like, because we talked about how nucleotides are interacting with one another, and all these are forming tertiary structures, and because of that, it's not going to look something like that, but in fact it's going to look something like this. But remember, this is the anticodon, and remember the anticodon is always not having any impact on the entire structure of the tRNA, and that's because the anticodon is just designed specifically to find its codon that it's made for, and then that allows the entire tRNA to take its let me just move that, to take its amino acid. It makes it a little bigger, resize, to take it's too big, take its tRNA to the designated area. That's the entire purpose of a tRNA.